www.nanohub.org.
You can follow along with this presentation using printed slides from the NanoHub. Visit www.nanohub.org and download the PDF file containing the slides for this presentation. Print them out and turn each page when you hear the following sound. Enjoy the show.
Hello again. So we'll continue our discussion on BioMEMS and Bionanotechnology. One of the things I wanted to start this lecture with is actually in slide 21 from lecture 3, which I was trying to show a video of dielectrophoresis, and for some reason that didn't work before. I think we got it working this time. So this is dielectrophoresis examples of, you can see how, what you see here is a real-time video of particles that are trapped within a interdigitated electrode array, and we have picked a voltage such that the small particles actually were going through. Now we increase the voltage, and now we can trap the small particles and the large particles. You can see that both types of particles are now stopped at the edges of the electrodes, or close to the edges of the electrodes. And then now when you drop the voltage, then the small particles can move again, and the large particles are still trapped. Okay, so this is an example of separation of particles with different sizes using DEP. Another example would be how you can manipulate particles and move them in different directions. What you see here is a real-time video of these little particles that are flowing from the left to the right. Hopefully you can see that in your video. And there's a channel here at the bottom and a channel on the top, and then electrodes are at an angle to the flow. And then when you turn the DEP on, then these beads, in this case these are polystyrene beads, they actually line up along these electrodes. They get pushed away from the edges and they are pushed to the top surface, and they can be moved along with the flow, but now at an angle within this potential well that is created in between the electrodes, in the center of the electrode. So you can take particles and move them in different directions from within a flow. Okay, and let me try this third one too real quick, see if this works. And here the idea is that you've got these particles coming in from the right, flowing to the left, and these are dielectrophoresis electrodes. So when we actually turn the voltage on, then these particles continue to get trapped at a very high efficiency while fluid is still flowing through this chamber. And in this case, the particles are all sort of lined up in this flow line because you have laminar flow, and earlier in the device they were diverted to the edge of the flow, to the edges of the chamber, and hence they stay in that flow line and they continue to get accumulated here. When the voltage is turned off in just a few seconds, then you will see that all of these trapped particles will then be released. Okay. So these are examples of dielectrophoresis that I wanted to show you earlier. Let me now move back to, move forward rather, to where we left off last time, which was on slide 50. Okay, so we finished up slide 49 and we were going to start on slide 50. So here, this is another example of a conductometric sensor that we have developed, which is the detection of bacterial growth and metabolic activity. So you can make silicon chambers, and that can actually be made within silicon or in PDMS or polymer devices. And you have these two electrodes. You measure the impedance between these two electrodes as bacteria is growing in this volume, and as the bacteria grows, then it actually changes the conductance of the medium that is growing in, and that is that change in conductance or the electrolyte resistance can be measured by measuring the impedance of these two electrodes. So that's another example of a conductometric sensor on a chip. Okay. Two of the types of sensors that I want to talk about briefly are actually what's called cell-based sensors, and these, I guess, don't directly fall into any one of these other three categories, but most closely they might be falling into the conductometric sensing category. But anyway, these are an important type of sensors, and they're called cell-based sensors. So here the basic idea is that the transductions of the cell, based sensors may be achieved by the measurement of the transmembrane and cellular potential, impedance changes, metabolic activity, analyte inducible emission of genetically engineered reporter signals, or optical by means of fluorescence or luminescence. So the basic idea is that if you take live cells and you actually culture them and grow them on an electrode surface, on some sort of a sensor array, And then the goal is to use these cells as sensors. Because at the end of the day, if you are looking for toxic chemicals or specific, let's say, gases that are toxic and dangerous, then one way to sense them is to actually use a live entity as a sensor. And a live entity could be a cell. So if you can keep cells alive, attach them to a surface, and then measure their activity somehow, monitor their activity, and when you actually put in a target species that impacts the cell, somehow destroys the cells or weakens their metabolic activity or slows them down, and somehow you can detect that using an optical signal or an electrical signal, then you really have a cell-based sensors, and that's what these sensors are. One of the main challenges of these sensors is to be able to grow these cells long enough for these sensors to be useful in portable conditions, in harsh environment over a long period of time. But there's been a lot of progress that's been made towards that front in the recent years. And the fifth type of sensor, electrical sensor, that I want to talk about is what we can sort of make a comparison to a Coulter counter. So a micro nanoscale Coulter counter type sensor. And the basic idea here is that of a Coulter counter really is that if you can make a small hole in a membrane such that you can only pass one entity, and Coulter counters, by the way, were originally developed for cells. So this pore needed to be many microns in size depending on the cell size. So if you can, cells can actually develop, or these different entities that you're trying to characterize, they can develop a surface charge. So if you put these entities in a chamber that is separated by this membrane with a hole, and then you apply an electric field across, then this charged entity would move, and the only way it can move through towards the other electrode is actually through this pore. So now what happens is that as the entity is moving towards the pore, before it gets to the pore, you could be measuring a background ionic current. So potassium or sodium or chloride ions, positive or negative, could be moving back and forth, and you could be measuring a background current. And then when the entity of interest moves through the pore, then it will actually block that background current, and the current will drop. And eventually when that entity leaves the pore, then the current will actually pick up again, as shown here in the bottom right. So this is really called a Coulter counter, this general idea, and this has been then applied on a micro scale with cells also, and also with DNA. So you can make these micro-pores for cells. If you can very easily make a pore that's a few microns in size or less, that it only allows a single bacterium to go through, and as the bacteria goes through, you can measure the change in the electrical current. You can characterize the electrophoretic mobility and the surface charge of the bacteria, and you can possibly separate live bacteria from dead bacteria that way. One of the most exciting examples of this idea of passing single molecules through pore has actually been done for DNA, and this has then become a very important area of research where various groups are working on making synthetic membranes with nanoscale pores to actually pass DNA through them. So this concept was initially demonstrated by Kasyanowicz and group using a lipid bilayer and a protein channel. So this protein called alpha hemolysin can be used as a channel. This is a protein-based pore that self-assembles into a structure with a 2.6 nanometer hole through it. Okay, 26 angstroms. And this is just barely enough to actually pass a single molecule of DNA through it. So what they did was they actually showed that you can measure the current through this pore, and then when you apply a voltage across it and put DNA on one side, then as the DNA molecule can actually stretch, and actually a single molecule can go through the pore individually, one at a time, and as these molecules are going through, you can actually measure then this drop in current. So the baseline is right up here on this figure on the right, and as the DNA goes through, the current actually goes down. Okay, and then when the DNA has left the pore, then the current actually comes back up. Okay. Now, what was also very interesting was this is really a size effect, right? Because you're really blocking the pore by the bases of the DNA, or by the DNA and its structure. So what was also very interesting and promising here, that they actually saw differences when you pass a region of molecule that has only C's versus only A's. Now, it turns out, if you go back and look at the structure of the DNA, that C is actually larger. C has 3 rings and A has 2 rings. So the larger molecule block the pore more, or the larger bases block the pore more, and the current drops a little bit more than when you actually have a poly A, or a region with lots of A's, because in that case the blocking current is a little bit less. I mean, the blocking is a little bit less and the current is a little bit higher. So that indicates that there is a potential in this approach to maybe someday sequence a single molecule directly, base by base. And that's really the holy grail for these type of devices, where the goal is that can you actually take a single molecule, pass it through a sensor like one of these pores with maybe some other sensors on the surface, and actually detect the passage of single bases as they're going by, and hence directly sequence a single molecule. So this type of sensor has been, there's been different approaches to make this sensor. I'll just quickly review this here, but the references are in the notes here, and you're welcome to go and look at them in more detail. One approach on the left here is in a silicon nitride membrane using argon beam, where you continue to etch this surface with a little cavity, and as soon as a pore is exposed, then this current through the pore increases, and hence you can stop the etching. So that way people have made these thin nanopore in silicon nitride membranes. And by the way, I should mention that the goal of making these synthetic membranes is very important, because the biological membranes might not be very stable under different harsh environmental conditions. If you can find a way to fabricate these sensors using top-down techniques that are more compatible with silicon device fabrication, then you can actually talk about making arrays of these pores and make sensors that could be actually handheld, portable, and useful. Another method that's been reported is using actually SOI wafers and silicon micromachining, where you can use take an SOI wafer and do some etching from the backside to create a diaphragm, and in this diaphragm then you can come from the top and do e-beam lithography and form this pore that is in the 30 to 40 nanometer range. And then once you oxidize this pore and then look at them in TEM, then due to the electron beam, the high energy electron beam that is used in the TEM, you can actually see that the pore size actually shrinks over time. And this is because of the fact that the oxide around the pore can actually flow and shrink to minimize the surface energy, and the pore will actually over time shrink and eventually close. And this is a very nice technique. It is a serial technique. You cannot do it in parallel. You do it on a pore by pore basis. But it's a nice technique because you can stop the process of viewing it, and hence control the size to whatever dimension you want. And there's some other interesting effects that take place here that I'll leave it in the notes here, but I won't go through them. So using this approach, you can also, we have measured the passage of single molecules through these devices. And there's some interesting effects that happen that actually are opposite to what people have reported. Typically, people report that your pulses actually go downwards. In these experiments, it was observed that actually the pulses go upward as the DNA goes through. And this was explained by actually a nanoscale electro-osmosis effect that might be taking place, where oxide surface have a surface negative charge, as we talked about earlier. They have these silanol groups which get negatively charged, and these negatively charged groups would have a positive ion accumulation close to the surface. Now, DNA has a negative charge on its backbone, so as the DNA moves through, it could be possible that it accumulates that positive ionic charge, and the current that's going through that mobile charge layer then increases, resulting in a net increase in current flow. Okay, so these are the 5 types of electrical sensors that I talked about: potentiometric, conductometric, electrochemical induced, and then the last 2 were cell-based sensors, and then these Coulter counter type approaches. So now I'm going to switch gears and move to optical detection. And it turns out that optical detection, I'm talking about it last, but it's actually really the most widely and most commonly used methods in microbiology and in biological detection and also in micro devices. Because of the use of fluorescence labels. So I'm going to talk about this briefly, but there's a lot of literature on the use of, fluorescence molecules or fluorescence markers to detect binding of DNA, binding of proteins, or even capture of cells, of course. Here is an example of an integrated optical detector that's done on a chip by Woden's group at Oak Ridge National Lab. And here the basic idea is that you actually perform an ELISA-like reaction of capture of E. coli cells on a surface, but then the detection or the indication of capture of the bacteria is performed actually integrated using these optics that are built onto the chip. So they actually have a helium neon laser with filters built into the device, and essentially you can do an integrated fluorescence detection on a chip. And they actually show here the number of E. coli cells versus the optical signal in millivolts. So essentially you can detect down as low as a few hundred cells using this approach with an integrated optical detector. Okay, this moves up to the last slide, so I'll quickly actually move to the last set of slides, which is really called it Lecture 3. But we have really taken about 5 lectures to cover these. Okay, so this is the last one. This is the last set of slides. And I'll actually now move to this area of microarrays, okay, and also tie that to optical sensing. So let's talk about microarrays and lab-on-a-chip devices. So optical detection in biochips can actually be performed using 2 main methods. One is fluorescence. Okay, this is the most commonly used method. Fluorescence molecules or markers are entities that emit light at specific wavelengths, and you can actually get enhancement or reduction, as in the case of FRET, fluorescence resonance energy transfer. Actually is a scenario where the optical signal is reduced upon a certain condition, and this can indicate, these approaches can indicate a binding reaction. Or you can use chemiluminescence, which is a generation of light by the release of energy as a result of a chemical reaction. This, by the way, just for the sake of definition, there's 2 types of chemiluminescence. One is light emission from living organisms. This is called bioluminescence, sometimes also called biological fluorescence. And the second type is light emission which takes place by passage of electrical current, and this is called electrochemiluminescence. Okay. So, and really the most common or the most common example of optical detection in biochips is these microarrays, which have an optical detection scheme. You attach a capture probe, and then you have a target probe which has fluorescent label, and you can look for this presence of fluorescent label, and the presence of the label indicates the presence of the target. Okay. So let me just give you a little bit more of an introduction to the DNA hybridization in microarrays. So these microarrays are, more and more used. They were originally developed by Affymetrix and Nanogen and then commercialized. There's 2 distinct approaches of making those microarrays, and I'll briefly talk about both of them right now. So these microarrays are really a basis for detection of unknown nucleotides and unknown genes or unknown short strands of DNA. And the idea here is that you want to do hybridization of an unknown fluorescently tagged strand with many known strands. So it shouldn't be there. With many known strands. And again, the reaction will determine the sequence of the unknown. So the idea is that rather than doing one reaction at a time between a target and a probe, can we find a way to make an array of your probes? So have an array with lots of probes attached, and then bring in your target and allow that reaction to take place simultaneously in parallel. Okay. So the strands, these capture probes, okay, the capture strands can be lithographically defined, as in the case of the Affymetrix approach, or electronically defined, as in the case of the Nanogen approach. and this can be defined at specific locations. So the basic idea is that you define an array of your capture probes, and then when you bring in sort of a target strand, if it binds to a specific site, then you can get a fluorescence signal from there. And once, if you get a signal only from one site, then since you knew the sequence of your starting strand, you now know the sequence of your unknown. That's the basic idea. So how can you place these probes at different sites? Well, one approach is electronic placement of DNA. So we know that DNA has a negative charge on the backbone, so why don't we use that principle and use electrophoresis to actually make an array? And the basic scheme is shown here. So you have a substrate which has electrodes, okay, which is some metal, and then on top of that there is an attachment layer. This attachment layer is pretty important, and it could be different for different schemes, but this, for example, could be a polymer layer that has avidin on it, okay, that has avidin molecules in it, such that the surface of the layer, this is, let's say, a hydrogel or a polymer layer that has these proteins called avidin, but then they're exposed on the surface, okay, also along with being inside, they can also be exposed at the surface. So now what happens is that if you apply a positive voltage on this electrodes, and you introduce on top of the chip, you introduce one type of capture probe, and you make sure that the capture probe actually have biotin at the other end, then due to electrophoresis, these molecules will get ionized, and they will move to the positive electrodes only and not to the other electrodes. And by the way, during this process, you can also apply a negative electrode, a negative voltage at the other electrodes to make sure that those capture probes don't go there, and you can only address a site you want to address. So once you have brought these molecules close, then the biotin at the end of the probe can actually react with the avidin that's exposed on the surface, and those molecules can then be attached to the surface. That's one way you could attach these molecules to the surface. And then you repeat the process. Now you have attached the first one. Now you go back and apply a positive potential on a second site, and repeat that process where you bring in a second capture probe, a different sequence probe, and get it to attach on a different site or a pixel, and so on and so forth. So you can continue this process, and you can build your own array. And that's really the main difference between these two approaches of electronically versus optically defined arrays. These electronically defined arrays, the user can buy these systems, the chips and the fluidic system and all that, and actually build their own array, whichever they like, where the entire sequence is being attached at the same time. Now, to continue that process, then what you do is now you bring in your target probe, which could be with a fluorophore, or I'm really showing that for simplicity case. Most of the time, you're actually going to have your target probe attached first. So let's say here is your capture probe. The target probe attaches to half of the capture probe, and then you have another probe which attaches to this, which has the fluorescent label, okay, where light is given off. So most always there'll be this scenario, but just for simplicity, I'm just showing the target probe having a fluorophore. When this attaches, now you let it react to any of these probes at the same time, and let's say you get binding on this side. Now what you can do is also you can actually apply a positive voltage on each electrode sequentially and get the molecules to come to that site much faster than diffusion would, right? Then they would move through diffusion if you don't apply a voltage. But if you apply a voltage specifically to each electrode in sequence, then you can bring the target molecules close to that site, increase the local concentration, and hence increase the reaction rate, and very quickly figure out whether you actually get binding or not. And then you can also apply negative voltage to push away the molecules that actually didn't bind. Okay, so you can use a combination of this scheme, and this is all software controlled, and you can, at the end of the day, get this array, basically, which will have lights coming off from different sites, indicating the extent of the reaction at those sites. Okay, And this is shown here. There are some pictures here from Nanogen's website. The actual chip here is actually very simple. All it is these electrodes on a surface with that attachment layer on top, and basically addressing these electrodes individually. They are working on essentially a row and column addressing scheme where you can then increase the density of these arrays. Okay, the other method is what's called the light-directed DNA synthesis on a chip, and this has been pioneered and then commercialized by Affymetrix. Here, the basic idea is that you are actually building the molecule base by base using a chemical reaction. Okay, and this process of building the molecule then is hard to do at the user site. So actually, the vendor does it for the user, and they actually work with you to design the chips for specific applications and pick the right sequences for your application, because the way they are built, they're actually built base by base. So the idea is that you take a silicon wafer, you have different sites, each one of these molecules represents a different site. You can attach these molecules first to the surface, and then what you do is you have a protection group. You have a protective group at the ends that you can deprotect by using light. So you have a photomask, just like you do a lithography step. You deprotect, let's say, one particular or two particular or X number of sites or pixels. Once you deprotect them, then you can actually perform a chemical reaction with individual bases to attach a single base there. So then, for example, you can put in a T only at these 2 pixels. Okay, then you can come back and use another mask and deprotect 2 other sites, and now perform a reaction with the C. Okay, so you do this. You will do a certain number of masking steps for each layer, and you will build the first base of basically all the molecules, and then you continue that process over and over again, multiple times, to build the molecules base by base using this approach. Okay, so it needs a lot of masking steps, but it can be automated and all programmed in terms of getting the masking done, bringing in the fluids, doing the reactions, and then doing the mask again. And these companies make automated systems to do that. So here is another example. For example, you on a chip, you can have this entire, in round 1, you can have this entire region with A's, and the next region with G and C and T. Then you can do a round 2 where you do it in the other direction, and so on and so forth. And you can continue to do it to really build up a combinatorial array of these probes. Okay. So the design of these arrays is actually very important, and in terms of what application you're after, what you're trying to detect, based on that you would design the array accordingly. Okay, so at the end of the day, what you get is this dense array, and then what you do is you look at the binding of your target with this array using fluorescence detection. So there is a fluorescence detector essentially within the system, and the fact that this is fluorescence detection, that will really ultimately will limit the size of the pixel in the array. Okay, so the current pixel size might be in tens to hundreds of micron, and the smaller you make the pixel size, the smaller, the weaker your optical signal becomes, because you have less fluorophores that will attach to that site. So ultimately, this will limit the size of the pixel in the array. And the applications here are numerous. Polynucleotide array for HIV sequencing, mRNA expression monitoring. This is really now, these arrays are used a lot for expression analysis. Here, I just want to make the point, remember that not all DNA reactions have to be perfectly stringent. 2 strands can bind to each other even if there's some mismatches there, right? So that's exactly what this array does. It actually also extracts out, looks at the intensity of the signal, and the intensity would relate to the concentration of the molecules that is attached, and that gives you an indication whether you have full, stringent hybridization, or there are even some mismatches. And the same concept can also be extended to proteins, although it is harder to make protein arrays because of the attachment chemistries of different proteins. But here, there's actually a nice example of an array that shows these dots of different proteins Essentially, in this case, the dots were still reasonably large. Here's a I believe this is a 1 millimeter distance, so each dot is still a couple of hundred microns. But essentially, in this case, you can also look for protein-protein interactions or small molecules interacting with proteins for high-throughput screening of chemical compounds. Okay. I also want to mention a note that when we talk about sensor arrays, you should keep in mind that really any of the individual sensors that I talked about earlier, the cantilevers or the nanowires or the pores or any of the sensors that we mentioned, they can actually be used in an array format to make micro-nano sensor arrays. Okay, so when we talk about microarrays, most often people tend to think these optical DNA arrays simply because they were the ones that were introduced first and commercialized first. But really, you can make microarrays with any type of sensors. Okay. And actually, people are working on that. So the sensors in the array need addressing, and each sensor can be then functionalized with different bioreceptor molecule to detect different entities. So you could have a cantilever array where each cantilever has a different DNA or a different protein attached to it, and that would be a label-free means of detecting, the biomolecular reaction. So examples include cantilever array, electrochemical detection in electrode arrays, cellular arrays for chemical detection, etc., etc. All right. Now let me move over to some lab-on-a-chip or integrated devices. So sort of taking these various concepts and putting them together to come up with examples of complete devices. And I'll go through a few slides here that talk about some of these examples. This slide shows an example of an integrated chip, a single chip device for DNA electrophoresis, okay, for DNA detection. So it's really a nice example of a device. This is made in silicon, where the chip was about 10 millimeters wide by many centimeters long, and the chip actually had these different regions. You actually have a sample loading region, okay? This was a silicon chip with a glass cover and with metal electrodes. You could actually load the sample and meter, meaning move specific amounts or specific quantities of the sample. In this case, they actually connected air lines to actually move the sample using a pressure-driven case to move the samples from, in terms of the loading and the metering, you could just move it using pressure-driven case. And then they actually perform some mixing in this region right here, and then there is actually a thermal reaction region where you could do PCR. So the idea is that you can actually move a sample that has some DNA and do a PCR on a chip right here. And the advantage of doing a PCR on a chip is that you can actually, because of the fact that you have a reduced thermal mass, you could actually get much faster temperature cycling. And you can take a small sample of DNA and then amplify it on a chip and then move this further down into this gel loading area and then actually run a gel right there on the same chip. And in this particular case, they also actually integrated photodetectors right on the chip, silicon photodetectors on a chip to get an optical detection using an electrical signal, of course. So this is a nice example of an integrated device that does everything from taking a raw sample of fluid which has some small DNA to actually doing a PCR reaction and then running a gel and detecting the presence of specific DNA. Another example of these microfluidic devices that I wanted to bring up is the CD formatted biochips. And this is actually another way to drive fluid through the chip. And I wanted to just mention it because it's a very interesting concept. And here the idea is that, can we use centrifugal and capillary forces to transport liquid? So this right here, what you see here, is really like a CD, really, okay? And the CD type platform, then you can use injection molding to define channels and wells. So these CDs are very, very cheap, as we all know. And then the basic idea is that as you spin the CDs at different velocities and at different times, then once you load your samples, then you can move the sample down or move this wash sample down, for example, or the calibration sample down by controlling the speed and the timing of the speed of when you change the speed of the CD. and you can actually perform the reaction and the mixing, and then you can also use the detection system for CDs, the optical laser detection system, essentially use that same system to detect the binding. So in that case, the long-term vision for the groups that are doing this kind of work is to actually do these chemical reactions and detection on one CD, and then use a CD player itself, for example, or the same electronics and the same optics to detect the biomolecular binding and the chemical reactions. Another example of a device for cellular analysis on a chip is to actually take, let's say very few cells or single cells. And this is a nice example from a group at Oak Ridge where they made these plastic PDMS chips using hydrodynamic, just pressure-driven transport of cells. Once you bring cells in a certain region, though, then they actually use electrodes to do lysing of the cell. So they did electric field mediated lysing of the cell, and once the cells were lysed and the products of the cell were released, then they introduced other fluorescent labels, and essentially then they had off-chip fluorescence detectors to detect the presence of specific proteins from those cells. Here's an example of a polymer microsensor and an actuator. And I should mention, actually, in this entire lecture series, I really didn't talk about actuators a whole lot, but there is a lot of work on making other components of microfluidic devices, such as valves and pumps, that could be integrated on chip and can provide a much higher level of functionality to do these biochemical reactions. So one example of that is using these polymer microsystems by BB's group up at Wisconsin. And the idea here is that you make these hydrogels, the kind of ones that I talked about for the cantilever. So you use similar hydrogels that are pH sensitive, for example, and you define them using standard lithography. And now what happens is that here, for example, you see a case where you have an inlet, and the outlet here is actually in the third dimension. So what's shown here in this schematic here, you can just look at this at the bottom left. So you have an inlet, and then the fluid goes up and then out in this channel. And right here, then there is this hydrogel plug that is placed. And now what happens is that if you flow a fluid which actually has a certain pH, then this polymer can actually swell. And as it swells or shrinks, vice versa, it can actually close or open that valve. So you can have a self-regulating autonomous system that can sense what's coming in and then close it, or close the valve or open the valve. An example here is this biomimetic valve using a bistrip hydrogel. So you actually have these hydrogel layers, these things right here, these flaps that can swell, and as they swell, they actually close. They become a one-way check valve where the fluid can only flow that way and cannot flow back. And when you change the pH down again to a certain value, then, or change the pH to the normal value again, then these polymers can actually shrink back again, and the fluid can flow through in both directions. Another example of capillary electrophoresis is shown here by Matthias's group. And the basic idea here was that you actually form these long channels for electrophoresis on a silicon wafer. So this is a silicon wafer or a glass wafer, but essentially you have these array of electrophoresis channels, and you can drop the sample at these, you can dispense the sample at these ports at the edges, and then again by moving this wafer around you can get the fluid to be transported. You have a common anode in the middle. Actually, in this particular case, you don't even have to move the wafer, but rather you put electrodes at specific sites. You have a common anode in the middle right here, and then you put the cathode in whichever channel you want to run, and essentially each one of these channels is to run the DNA through or move specific chemicals through electrophoresis. Another example of work that we're doing is this idea of integrating different functions that are needed for detection of bacteria or cells all on a chip. So you like to be able to do some concentration and sorting. You could do that using electrophoresis. You like to be able to capture selectively the cells of interest. You could do that using antibody-based capture. You could do a viability detection of cell growth. Then you could lyse the cell and then look at the products using label-free sensors like the cantilever, nanopores, or nanowires. Here's further examples of microfluidic devices made in polymer. So this is a polymer device on silicon, actually. That's a hybrid device. Here's a region to dispense the fluid, a little reservoir, that then you can drive that fluid through the channels using this tube. One of the issues with PDMS is the fact that it is a porous material and fluid can actually evaporate through it. So if you have very small channels and you or very small chambers and you want to keep that fluid and use it over a long period of time, then that's always challenging. So there's some results here where you can use different materials by changing the composition of the PDMS and making it stiffer, and then you can increase that evaporation time of the fluid from a few hours up to 20 hours or so. Okay. So that really sort of covers all the topics that I wanted to talk about in this lecture, and I'll give you a couple of final concluding slides. So the future directions in this area is really, this is a very exciting area, and where things are going is in these following directions. So the idea of integrating more and more sensors and more functions onto a single device is something that we are more and more interested in. So integrated devices for analysis of single cells. This can be used for many applications and also for some fundamental science for studying the functions of single cell, and being able to probe single cells would be very useful for basic biology. To be able to use micro and nano fabrication techniques, techniques that actually are inspired from micro nano fabrication, soft lithography, bottoms up and top down fabrication, but then actually building biological structures using biological entities like proteins, DNA, and cells to build cell by cell, or to do tissue engineering using micro nano fabrication. That would be very, very interesting and very useful. To be able to integrate diagnostics and therapeutics. Again, this is one topic that I didn't really talk about, but using similar type of devices, you can also talk about therapeutics, where you're talking about releasing specific amounts of chemicals and drugs to the patient. So the whole idea of controlled drug delivery or intelligent drug delivery, but then integrate that using these micro devices, integrate therapeutics with diagnostics, so you can sense and release. And then also tools for genetic manipulation and characterization of single entities, microorganisms, cells, and viruses. Again, this sort of links back up to the top, this first one, but this whole idea of synthetic biology, where you want to be able to modify the genetic code of specific microorganisms for different functions, and to be able to do that at the individual particle level, then you will need devices like the BioMEMS and Bionanotechnology. So that brings me to the end. I'd like to really acknowledge my group that helped me with generating all the material that I presented, and also our funding agencies. We have in our research group funding from USDA, NASA, NSF, and other agencies, also our faculty collaborators. And I'd like to then especially thank the NSF Center for Computational Nanotechnology here at Purdue, that actually is responsible for videotaping these courses and then making them and turning them into the right format to actually put them on the web and disseminate this out all to you. And that was my last slide. Thank you very much. And if you have any further questions about this, you're welcome to send me email at bashir@purdue.edu. Thank you, and have a good evening.